Hello, my name is Jalen Cox, and this is my exam mini video number 4 concerning the second prompt, which was the transcription in prokaryotes. The transcription in prokaryotes, there's an important part of transcription is that the RNA polymerase enzyme starts off as a core enzyme, but through the sigma binding factor turns into an holo enzyme, which is used in transcription. There are 3 different processes through this transcription, or 3 different steps, being initiation, elongation, and termination. So step number 1 is initiation. So RNA polymerase binds to the promoter region, and DNA begins to unwind. The holo enzyme is required to bind to this promoter, and there are 3 different regions in this promoter: the -10 box region, the -35 box region, and the +1 box region, where initiation actually begins. And I've created a little visual there to better understand just the structure, of initiation. So in here, I'm going to answer this question. I've recreated my last thing, but I'm going to use it to answer this question. What if we increase this region by 5 BP or 5 base pairs? And I answered it this way: if the order of or number of bases were to be changed, then mutations could occur, which could change or stop the production of essential proteins. For example, this mutation could change how the genes are coded, resulting in an organism who cannot produce the necessary proteins for replication, or even new muscular tissue, and they would probably not survive. Step 2, elongation. This is the second step. So there are 2 strands, the coding and template strands, and RNA polymerase moves to the template strand from 5' to 3' and produces RNA transcript. And there's another visual there. And just remember that it always moves from 5' to 3'. And the final step, the third and final step, termination, is where RNA reaches the termination site, and the RNA transcript is set free from the template strand of DNA. There are 2 different ways that this can occur: in the Rho dependent, which uses the Rho protein that binds to RNA and uses helicase to separate the DNA and RNA chain, and Rho independent, where there is no Rho protein and uses a termination hairpin made by inverted sequences. The chain, this Rho independent chain, also ends when there is a long sequence of adenine at the end of the nucleotide. And here are my sources for this module video. Thank you.